Hi, everyone. This is Mrs. DeWitt, and today we're going to be looking at this coloring book page called The Action of Hormones. And so we're going to be looking at the difference between protein hormones and lipid hormones and some of the different responses that hormones can initiate once they're inside of a cell. So before we get started, you'll want a regular pen or pencil, a highlighter, and the color penciled colors that you can see here. And so we're going to get started. So hormones. All right. Hormones bring about chemical changes in the body by modifying the metabolic activities of their target cells. So remember, the word metabolic stems from metabolism, and metabolism is just all of the different chemical reactions that happen inside of your body. So hormones can start or stop chemical reactions in your body and their target cells and tissues. Although there is still much to be learned about the mechanisms of hormone activity, scientists do know that different kinds of hormones act in different ways. For instance, steroid hormones are lipid soluble and enter the cytoplasm of the target cell where they bind to receptors. Remember, steroids are actually lipids themselves. That makes sense. Like dissolves like, the lipid bilayer allows lipids to easily pass through, even big ones like steroids. But protein and amine hormones, so amine just means like really tiny hormones made of a few amino acids, cannot diffuse through the cell membrane. They're polar. And so they combine with receptors on the surface of the cell membrane of their target cells. So I would just star that section right there. That is essential to remember, and hopefully you remember that from one of our first units of the year, where we differentiated between different macromolecules. In this case, remembering lipids versus proteins. In this plate, we will examine both of these mechanisms and study other aspects of the methods of hormone action. Bold colors, blah, blah, blah. All right, we're going to see that there's two diagrams that reflect the two pathways of hormones. We're going to start with the first one called the mobile receptor mechanism. And we're going to kind of go back and forth between what we're reading here and the diagram. So I'm going to put down my highlighter and I'm going to pick up my yellow colored pencil. And throughout this, we are going to color code. It's kind of like that golden yellow color. We're going to color code steroid hormones as yellow. So steroid hormones act on their target cells by a mechanism currently known as the mobile receptor mechanism. In older texts, the process is also called gene activated mechanism. And this mechanism is so named because the receptor for the hormone becomes mobile after the hormone binds to it. So that is going to start over here, mobile receptor mechanism. And let's just go ahead and start by coloring. This is our steroid hormone, and we can label it steroid. And underneath, I'm going to have you write lipid to remember that it's a lipid. So testosterone is an example, estrogen is another. And so again, this is the mobile receptor mechanism. And the main idea is that steroid, again, our lipids, signal molecules like hormones diffuse through the membrane. And we can see that with this arrow right here. And we're going to read about this next part that's going to happen inside the cell in the next paragraph. So the process begins when a steroid hormone leaves the bloodstream, and approaches the cell membrane, which we know is a lipid bilayer of the target cell. The hormone passes easily through the membrane because steroids are lipids to enter the cytoplasm, where it combines with the receptor. I'm switching to green. And we're going to use green for this intracellular receptor, which means on the inside. This receptor is a protein, okay? And the union of the hormone and receptor creates the hormone receptor complex. So we've seen stuff like this before. This is similar to like an enzyme and its substrate fitting together. In this case, it is a lipid and a protein that fit together to form a complex. So let's go check that out in the diagram here. So here is our intracellular receptor. And again, intracellular meaning on the inside. And we can see it's mobile, which is the name of this. When they bind together, this can move around through the cytoplasm, and we're going to learn about where it can go next. But let's go ahead and label this in the meantime. It's labeled D, intracellular receptor. Okay, so the hormone receptor complex that we just colored, and we can color code it again. Again, it is this special complex has a hormone that is made of a steroid, which is a lipid. Even though it's attached to this protein, which usually isn't lipid soluble, as a complex with that lipid with it, is able to enter the nucleus, which is really special because, as you may know, we don't let a lot of things in and out of the nucleus because the whole job of the nucleus is to protect the DNA. And so this hormone receptor complex is able to do that. And we know the nucleus is surrounded by a nuclear membrane and has nuclear pores. And so where DNA comes into play here is that this hormone receptor complex actually binds to the DNA in the nucleus, and it can activate one or more genes. Now, we're going to learn way more about this later in our gene expression unit. So this is kind of just your first look at it. So this gene activation is the reason that the alternate name of this mechanism is sometimes used. The activation induces DNA to encode a molecule of messenger RNA, mRNA, and regularly occurs as the mRNA molecule migrates to the endoplasmic reticulum of the cell, where it instigates the synthesis of a protein. This protein brings about the desired effect in the cell. For example, the protein may act as an enzyme that alters the rate of a specific cellular process, or it may act as part of a membrane transport system to alter membrane permeability. So basically, we're talking about protein synthesis here. That isn't always going to be the response, but these hormone receptor complexes can go in the nucleus and they can turn genes on, meaning we're going to make the protein that gene codes for, or the opposite can happen. We can turn a gene off and we stop making the protein a gene encodes for. Let's go ahead and highlight some of the other important information in this paragraph that we just read. So again, the complex itself bound to DNA, it activates one or more genes. And like I said, in some cases, we can deactivate genes. And when we're activating, we create mRNA that can then leave the nucleus, go to the endoplasmic reticulum, go to a ribosome specifically, do translation, and then we get a protein that can do stuff. So you hopefully remember this as protein synthesis. But basically, we are initiating the synthesis of a protein. So again, protein synthesis. And down below, again, you basically are turning genes And again, genes are found in the DNA. They're little chunks of DNA that code for proteins. Oops, sorry about that. They are turning genes, which are found in our DNA, on or off. And this is called gene expression. And again, we're going to have a whole unit, a whole packet just dedicated to gene expression, and it's pretty cool. And when we express a gene, usually that means we're making the protein that gene encodes for. So gene expression is how we do protein synthesis. You'll see these used interchangeably a lot of the time. So let's go ahead and finish that off over here in our drawing. So again, we made the nucleus purple. So we're going to we won't color the whole thing in. We'll just shade this outside part. And maybe you remember a little bit about this from your introductory biology course when you were probably in 9th grade. Okay, so what we're looking at is a little piece of DNA here. And all along the DNA in little chunks, hopefully you kind of remember A, T, C, G, you have pieces called genes. So we're going to kind of just draw a chunk out of this. I'm going to say from here to here, let's say is one specific gene that codes for a specific protein. And we'll just shade that in purple too. And we can turn this gene on, and the gene right next to it can stay off. So it's like flipping a light switch, right? When you flip one light switch in your house, unless it's a master switch, not every single light turns on. That would be crazy. So you can turn off individual lights, you can turn on individual lights, and that same thing in your DNA. You can turn on and turn off different genes for when you need them and turn them off when you don't. So again, this is a gene right here. And we see that the hormone receptor complex is responsible for kind of triggering this event. And so in this case, this hormone is an activator. You can come in and activate the process of transcription, which again, we'll review, but transcription is when DNA goes to messenger RNA or mRNA. We are transcribing the message so we can do that first step of making proteins. So in this case, we have an activator. We're going to activate this gene, and it's gonna turn to mRNA, which we can see right here. So H is mRNA. Okay. And that mRNA is a transcript of the DNA, but only that gene. So we call this an mRNA transcript. And maybe you remember that this mRNA can leave the nucleus. And here we see it being translated into a protein. And so we can see that it has the altered cell response. And in this case, it was protein synthesis, which is the whole goal of why we did this in the first place. All right, so again, protein synthesis, let's just go ahead and highlight that. This was the response, that the whole reason we sent this little signal over here in the first place. And so the nucleus takes part, we activate the gene, it gets transcribed to mRNA, and mRNA leaves, finds a ribosome, which is represented by all of these tiny little bumps. So we can say ribosome. The whole thing is the rough endoplasmic reticulum. And we're doing protein synthesis, we're gonna make a protein. And so this is our response. And in this case, the response is gene expression. We are expressing a gene, turning it on in this case, and making a protein. So that's one example of a response. So One thing that I've taught you so far is that in cell communication, when we actually get to the cellular level, we can break down cellular communication into 3 steps: signal, transduction, response. And so in this first example, this is our signal, and we receive that signal when these bind, when our receptor and our signal bind. In this case, a steroid lipid, and in this case, on the inside of the cell because this is lipid soluble. Transduction is a little different. We have gene expression, but basically we are going in, there's going to be a series of events that happen that lead to transcription and translation, and we get gene expression. This last part, actually getting gene expression and having this protein being made, is the response. Now we're going to see a kind of more common example of signal transduction response. And so we're going to move over to that next section now. Okay, so the next type is when our receptor is on the outside of the cell, extracellular. So many protein and amine hormones act by combining with specific receptors that are present on the target cell membranes. And after the union has occurred, the receptors remain on the membrane. And so we know that proteins cannot pass through the lipid bilayer. And so they need these extracellular receptors to pass on their message. So the first step in this mechanism occurs when a protein hormone, and we are going to use red, approaches the cell membrane, where it unites with a specific receptor. And we're going to do blue for our extracellular receptors for this model today. And it's going to be located embedded in the phospholipids of the lipid bilayer, and it's going to be kind of sticking out. Sometimes it's kind of flesh. They're all a little bit different. The union of hormone and receptor has an effect on the enzyme adenylate cyclase, which is bound to the other side of the membrane. I want to pause for a second and note that this is one specific example. It's a very common example. There are some other ways that this happens. This is a good example to really get to understand those. It is very common. So again, adenylate cyclase. In other texts, you might see it called adenylyl cyclase, basically the same thing. And so adenylate cyclase is bound on the other side of the membrane. Normally, adenylate cyclase is inactive, but this union between hormone and receptor changes it into activated adenylate cyclase. The activated adenylate cyclase catalyzes the breakdown of an ATP molecule. And so we're pretty familiar with that. But in this case, we have another ATP friend called AMP. In fact, cyclic AMP. Cyclic AMP is maybe what you're thinking of, okay? This is adenosine triphosphate. Oops, hold on one second. All right, my iPad froze for a second. Okay, ATP, we know, is the energy of the cell. It stores and releases its energy in the 3 phosphate groups attached to it. We're very comfortable, hopefully by now, with ADP being the uncharged battery. We add that 3rd phosphate to it, and we have ATP. Now we're introducing cyclic AMP, adenosine monophosphate. It's cyclic because when it only has this one phosphate attached, it kind of curls around and forms this circular shape, cyclic AMP. We call it CAMP for short. So cyclic AMP acts as the second messenger in this series of reactions. You're going to hear a lot about second, and sometimes they're called secondary messengers in these signal transduction pathways. They're kind of the connection between reception and transduction. And so they're the ones that kind of activate what's going on inside the cell. This hormone is considered, again, the first messenger. So the protein coming in, and we're going to go over here to the picture now, is that very first messenger, but it can't cross the cell membrane, again, because it is made of protein. A very common protein hormone we're going to talk a lot about in this class is insulin. So as we know, insulin helps control our blood sugar and get sugar out of our blood and into our cells so that it can be used. And so this is a protein hormone. Protein hormones are often called ligands, or depending on the person saying it, sometimes it's pronounced ligand, and it's still a hormone, but it cannot cross the cell membrane. So it binds to this extracellular receptor. First messenger, second messenger is adenylyl cyclase. At this point, it's inactive, and you're going to see that with this reception, it activates, and now transduction has officially begun. And again, transduction is this whole chain of events that's going to occur inside the cell in order to finally get to a response. So it triggers, I like this part, a chain of events in the cytoplasm that constitutes eventually the cellular response. So this paragraph goes a lot over what that transduction is like. The mechanism continues with CAMP. CAMP activates an enzyme called protein kinase. We're going to green again here. Green means go. Okay, so this is an activator. So protein kinase in turn incites a specific event. For example, protein kinase activates enzymes in the liver to alter the rate of glucose metabolism, and also acts to reduce tension in smooth muscles, increase the rate of contractions in cardiac muscle, and stimulate the secretion of thyroid hormone. So protein kinases are activators, they are goes. We're going to go ahead and color that over here. And we can see that adenylyl cyclase took ATP, turned it into cyclic AMP, CAMP, and then CAMP, with the help of a protein kinase, is going to turn on our target cell responses, which could be any one of those things that I just said. We can be, right back over here. We can be altering the rate of glucose metabolism, increasing it or decreasing it. We can reduce tension in smooth muscles. Any of these things, we are stimulating a response of some sort. But we don't always want this on. We eventually want it to turn off, and that is this next section. If cyclic AMP remained active too long in the cell, the chemical changes would be disastrous. We'd be doing the same thing over and over again. So maybe the response was to break down glycogen into glucose because we need glucose. But if we kept doing that nonstop, we would have no more glycogen and we would run out of all of our energy storage. And again, that would be disastrous. We would have no more energy in our body. That'd be very dangerous. So we want to have an off switch too. So the CAMP must be changed back to inactive CAMP, or ATP basically. This change is brought about by phosphodiesterase enzymes. You can pause the video or replay that back, practice that. Ready? Phosphodiesterase. That one's fun, right? And again, both of these end in ACE, so we know they're enzymes. This inactive CAMP then passes out of the target cell and into the bloodstream. Now the receptors at the membrane are available once again. And so where we have our phosphodiesterase is over on the side here. We'll color code this red. And we can deactivate CAMP and we can turn it off. Off, so that we're not stimulating this response over and over and over again. Now I want to tell you a little bit more about protein kinases and these phosphodiesterases. So protein kinase and these are vocabulary terms that I do recommend you work with and try to memorize and look up it in different ways so you really understand them. And what they do in order to turn things on is they add phosphate groups. So they add phosphates to turn on. And so we're going to color code that green again. And then I'm going to teach you my little way of remembering this. So kinase, I like remembering connect. We are connecting phosphates to turn on. It also kind of sounds like kinetic energy, which is just the energy of motion. So we can think of on, we can think of energy. So protein kinases connect phosphates. So connect on. So protein kinases turn things on by adding phosphates to them. Phosphates carry a large amount of energy. That's why ATP has 3 phosphate groups. So we're turning things on, we're energizing them with protein kinases. All right, now, this term is not used that often. So I'm going to give you the term that is used more often, especially in the world of AP biology. Phosphatase. Okay, so phosphatase, a little bit easier to say than phosphodiesterase. This removes phosphate groups to turn things off. So these inactivate other molecules, inactivate enzymes. And I have a helpful way to remember this one too. At least I think it's helpful. I hope you do too. So phosphatase. Here we go. Phosphatase. So I'm going to say take it off or take it away. I like even better. But you can write both. Okay. So protein kinase connecting on. Phosphatase is take it away. Take it away. So you can just remember take it off or take it away. And we are taking off phosphate groups, which are removing energy. And again, if you are comfortable with the ATP, ADP cycle, we know that when we break that third phosphate group off, it releases a large amount of energy, but we're going back to ADP, which doesn't have as much energy. So we are taking phosphate groups off, and that is reducing the amount of energy in the molecule, okay? So back to our diagram over here. Let's remind us where we started. We had a protein hormone, can't pass through the membrane, so it binds to this extracellular receptor. Then transduction begins here, okay, with activation of adenylyl cyclase. We have a whole chain of events. That is what transduction is, a signaling cascade. We have this protein kinase turning things on. We get our final response. So that response doesn't just keep going. We have a phosphatase turn it back off. So in our 3-step system of signal transduction pathways, right up here, where this is locking into place, this is step 1, right? Reception. Boom. I'm gonna grab my highlighter here, and transduction is these steps right here. And we'll even include the off part. Okay. All these steps, this chain reaction that had to happen was step 2, transduction. And transduction is often referred to as a signaling cascade. And a cascade is a chain reaction. It's a chain of events. So it's kind of like playing telephone, if you will. And then last but not least, we have our response, which is a cell function of some sort. Okay. And so when we are comparing these two different types, one thing I want to make very clear is they don't always have to match up like this, okay? Usually we have steroid hormones passing through, and then these are the ones that can pass into the nucleus and act as transcription factors and make protein synthesis happen, gene expression. However, there are some proteins that can trigger gene expression as well. And there are some lipids, hormones, that can trigger a cell response. It can trigger a regular reception transduction response, signal transduction pathway. So these responses can be flipped, and these proteins and these steroids can be flipped. So it's just keeping in mind that this is one example of these two different main ideas. And learning a few examples really well will be really helpful, because on the AP exam, you never know what you are going to get. But in general, we have our hormone coming in. Proteins can't pass through extracellular, steroids can intracellular. This is reception. So just like we had reception here, we can label it here, reception. But the difference is reception, I almost forgot the I, with steroid hormones can happen inside the cell, intracellular receptor. Transcription and translation can kind of be considered like transduction, a little different, but we get our response, gene expression, okay? Transduction, this is a really good example down here, where we go from one to the next, to the next, to the next, and we can turn it off. So I hope that this was helpful in getting into some of the details of signal transduction pathways, the different ways that hormones can interact with receptors, and the different responses that can happen as a result. And please feel free to go back and rewatch any part of this video. And that's it for now.